Today, for the second talk, we're going to be talking about tests and diagnostics, looking at how these are implemented and what they offer. And then after this, we're going to go into early diagnostics for breast cancer with IVDs. So the first talk was a little superficial. This next one is going to be kind of in the medium length, and then I hope you stick with me for the last one because that's like a full-blown science conference talk, and I think it's important to have that as well. So yeah, here are some quotes and some studies, everything from the Cochrane Review to the journal, the New England Journal of Medicine. These show why tests, and especially diagnostics like ours, are so critically important. Patients equate testing with thoroughness, and they want a thorough doctor. They view doctors who order more tests as more competent, right? Really, back in the day, a good doctor would be able to tell you what you had without even a single test. It's a little counterintuitive, but this is the perception. This is what this publication in Inhalation Toxicology showed. In a 2006 review published in the British Journal of General Practice, showed that 90% of people want their doctors to offer them at least all tests possible. I'm in the 10%. 85% want to make decisions about their healthcare with their doctor. So obviously a report like ours that shows actionable next steps derived from data that's identified is incredibly useful. So patients obviously view diagnostic testing and this collaborative decision-making process with their doctors as being incredibly important when they choose who is going to administer their care, and is again a reflection of the provider's competence, right? In a 2009 publication, the Cochrane Review looked at 86 total trials, and they found that, here, this is an actual quote, everything that's underlined is a direct quote. The use of patient decision aids for a range of preference-sensitive decisions led to increased knowledge, more accurate risk perceptions, a greater number of decisions consistent with the patient's values. We do that in certain cases when we are taking in survey data, and a reduced level of internal decision conflict for patients. This is a huge thing as you guys are going out there, and thankfully we don't have to deal with that as the poor hungry doctors. And of course, fewer patients remaining passive or undecided. So what are we going to talk about today? We're going to talk a little bit about metabolomics and diagnostics. We're going to go a little bit into some case studies. We'll talk about integration into practice, although this is a very brief section of this talk. Gail is going to be leading that mostly. And then we'll talk about digital twinning, some precision protocols, and then leave some room for Q&A. So first of all, what are this is precision medicine, right? So you were talking about this cancer treatment, this chemotherapy that works in 30% of people, and they know that before they administer that. And they know that before MD Anderson takes your $100,000 deposit. Our goal with personalized treatment, of course, is to have more people have the expected effect, reduce low effect, and as much as possible, completely wipe out this adverse effect that is possible. So we have this multidimensional AI algorithms, and these can help us do really unique things. We can identify nuanced biomarkers. We can integrate that with patient histories, as we do with our Omni survey to the best of our current data collection abilities. We can use this to, of course, look at radiographic images, and of course, integrate this with wearable technology that makes sort of health prompting and real-time monitoring possible. So for instance, a really important thing is that the Yale New Haven hospital system reduced mortality of sepsis patients by 29% by using the Rothman Index. Have you heard of the Rothman Index, right? It's a combination of some vitals, some clinical laboratories, and even nurses' notes, right? And it creates this very easy to use quantitative handle that kind of lets hospital staff know how critical this individual's case is. In fact, Epic software systems, right? They now have a built-in Rothman Index AI that does sentiment analysis on the nurse's notes, and it can quantify the sentiments expressed in the nurse's notes and includes that as a more accurate calculation for their Rothman Index. So these are really useful things that have already been utilized dexterously to some appreciable benefit. So this is the science of precision diagnostics, right? And you can see the legend down here. It says submitted to biomedicines, but as of last night, it's been accepted. So this will come out soon in publication, and what we are doing is, okay, so we are in the laboratory, we take our diagnostics, we analyze various therapeutics from digital twinning. Once we have that mapped, we've mapped their biology and we've simulated the effects. Now we have personalized medicine, and then more data collection from metabolomics at retest. This goes into the data management, then we can generate some hypotheses between tests, and then it goes back into the molecular epidemiology, and this cycle repeats. And the more iterations of this cycle we get, the more precise we are at recommending these treatments that are predicted to move the needle for this individual. The more we learn about them through longitudinal testing, the more accurate these algorithms become. So we go from analytics, we go from data generation to analytics, and the end goal is this actionable knowledge, right? There's a famous quotation by Daniel Raftery. He's one of the founding fathers of metabolomics, and he says that we have, seas of data. We have, pools of statistics. We have lakes of findings, and then he says we have the odd drop, or no, we have lakes of knowledge, but the odd drop of wisdom. And so that is what we're trying to distill here with the science of precision diagnostics and molecular testing. So metabolic biomarkers, metabolomics is inherently a comparative science. We can start with cell lines, as you saw in our cell room. We can start with animal models, as we have downstairs, or clinical trials, as they're run on this floor in the mock clinical room. We take these biospecimens, we use either mass spec or NMR, we detect and quantify these metabolites, those get shot out as biomarkers, we do some statistical modeling, and throughout this data collection and modeling process, we are feeding information back into the mechanism. We are postulating mechanism. We can prove mechanism with metabolomics if we do this other technique known as flux analysis, whereby we can label somebody, or if it's in cells, we'll just put some carbon-13 with the media. We'll track how that goes through the TCA cycle because carbon-13 weighs just a little bit more than carbon, and it's only 1% of the naturally occurring isotope of carbon. So it's not in the environment. If we feed it to somebody, we can track the carbons as the heavy carbons go from molecule to molecule through a pathway. And if we sample something like a petri dish through time, we can tell what pathways are lighting up, and it's a dynamic look. It's not snapshot metabolomics. We do some statistical modeling, and then we kick out some systems biology information. This is the general breakdown of metabolomics analysis, right? And as we are, of course, we talked about metabolites being less than 2,000 Daltons. Really, it's less than 1,500. We are downstream of the genome. That genome is being transcribed to RNA, translated to protein, and we have two really sets of metabolomics proper. We have the aqueous metabolites, which is metabolomics, and then we have lipidomics, where we're able to generate data on hundreds of lipids in a single run. So this is, of course, the central dogma, and we have the phenotype. This is a really interesting scenario where sometimes we test an individual. We had an individual test with us. He was quite literally on the last month's cover of either Men's Health or Iron Man magazine. He's this amazingly fit person. And he scored really poorly on our test, really poorly. We were worried. And he had some ideas about what it could be, but essentially his phenotype did not match his metabotype. And this is something to talk about, right? We know that if somebody has a certain genotype, there's no guarantee that they will have that phenotype. You can have the 6 or so really high variance accounting SNPs for schizophrenia and never express schizophrenia if you're not in an environment that elicits that. So your genotype and phenotype don't match. And the same thing happens with metabolomics. Oftentimes we'll test an individual and the metabotype doesn't match the phenotype. Now it's either because the metabolomics is telling us, reporting on really sensitive changes that are happening minutely that still have not been realized at the phenotypic level. Right? If the mass spec does not lie, if you do not understand something, do more thinking. Or oftentimes it's the other way around, where a person feels very sick and their metabotype is normative or even at times optimized. The issue then is the metabolomics is not the problem. That's the conclusion. Unless we're doing multi-omics, then for that instance, that patient who is fermenting sucrose from fruit and generating aldehyde metabolites, this is not the issue. We see this and we think, oh, they might be having too much of the sauce. The issue is the microbiome, which is out here and is modulating all of these different variables. So why don't we use NMR? NMR, nuclear magnetic resonance, I've only used it once in a single paper of mine, but it has great coverage, right? Like when we look at metabolites in a three-dimensional space and we sort of plot them with regard to their hydrophobicity, their acidity, and their concentrations, NMR is pretty good. It can handle almost all types of metabolites, but it has to be a high concentration, or else NMR really falls apart. It's quantitative in nature, but doesn't have the low concentration. So MS is 10 to the 6 times more sensitive and more selective. It can be less reproducible. So whenever we validate or create a new panel, we always validate with standards. And if we're comparing results between studies, we have certain correction factors that account for different instruments or columns that are used. NMR is far more sensitive. So we have various types of mass spec that we try to make more usable and more reproducible. We can do that by doing things like LC-MS to the N with ion traps that, again, can take that base peak mass to charge ratio, split it apart, and look at satellite peaks. You can have two molecules have the same molecular weight, and again, they might have the same chemical formula, but their arrangement in space, in three-dimensional space, is different. And because of that arrangement, if we split that base peak apart, we will get varying satellite peaks, which can be used to identify a metabolite with more confidence. We have ambient mass spec for things that are kind of, they're not amenable to hard ionization techniques like EI. What we use is hard ionization. If we were looking at cocaine, for instance, we could not use EI. It splinters the molecule too much, and the spectra is just cropped, and it's not amenable. So we would use something like ambient mass spec that kind of, instead of hitting it with an electron beam, we take a gas chamber, we ionize that gas, and then we pass your sample through that gas, which is an ionic cloud, essentially. So there's soft ionization, hard ionization. This is what we use. This is what you guys saw in the lab, right? You saw it in person. So you are the source of metabolites. We take a dry blood spot. We do grinding or homogenization, extraction. We then derivatize those molecules. We make them amenable to gas chromatography analysis. We put them on the chromatography column. They all come in the same, but they separate in that column as to fast eluting, denoted by the blue dots, or slow eluting, denoted by the red dots. And as this is coming through, right, so you have your gas inlet, as we were explaining. Here's the, let me see if I can. Here's the gas inlet coming through. This is your sample. As it's coming through the inlet, it is hit with this electron beam, and then it is going to be ionized. It passes through a series of acceleration lenses, and then it, essentially using radio frequency and AC/DC, we are able to filter metabolites. So you will get a chromatogram, and if we take a slice of that chromatogram, you will see the mass spectra, right? So metabolites correspond to a certain retention time and a mass spectra. If we slice that chromatogram open, and that's what you saw on the screen being collected was the data. You saw the chromatography peaks, and then you saw the mass spectra peaks being generated at every second. Okay, so we've seen this before, and I won't spend too much time here, but these are the 80 pathways of Homo sapiens from KEGG. Why is KEGG important? And this is something that Alex brought up on the page. KEGG is a standardized database that allows researchers to reference certain pathways and be sure that we're talking about the same thing. If I say mitochondrial, if I say TCA cycle, and you start talking about let's say an anaplerectic amino acid like glutamine. Glutamine is technically not a part of the TCA cycle. It is related to the TCA cycle, but it's not TCA cycle proper. So when we use KEGG, we are basically saying we agree that these are the 80 pathways. And KEGG is updated from time to time, not as much as it probably should be, but it's updated from time to time by the Kanaisa laboratory in Tokyo, in Kyoto. And so this is the 80 pathways that monitor that are involved in KEGG. And of course, here are the 68 pathways that we cover. And then this is the coverage by the CBC, BNP, lipids, liver function test, thyroid function test, and an OATS test. Okay, so we are seeing more data. That should be obvious. And what should be obvious from our talks prior to this is that there's no way you're going to analyze this data, not unless you want to spend, a day or two really going over this for each patient. This is not a sustainable way to do medicine, especially when we have high throughput techniques like bioinformatics pipelines, and we just press a button and everything's kind of calculated and issues are identified and protocols are generated. So here's one person who we had a case study of. John's a 42-year-old male. When we looked at his metabolomics, so John was a special case where we actually did short chain fatty acid analysis too. We have a panel that we haven't commercialized yet, but it looks at 14 short chain fatty acids. This can be applied to blood or it can be applied to stool, as Alex was alluding to our upcoming microbiome test, where we're going to look at sequence your microbial bacteria, but we're also going to be looking at the short chain fatty acids that they produce. And these 14 short chain fatty acids are key to understanding microbial communication. So he was a difficult case. Our first test did not run well with him. So we took his second sample that we had already on hand and we ran a short chain fatty acids panel. What we saw was severe dysregulation beta oxidation. So the pathway through which these fatty acids are burned through the mitochondria and generate, of course, ATP, we saw that this was incredibly low, totally throttled in his case. So, of course, we gave him some dietary interventions, and those dietary interventions basically were, consume more malic acid, have some alpha-lipoic acid. These are, we also wanted to increase the number of his mitochondria, so we gave him a little bit of CoQ10. The alpha-lipoic acid was the main protagonist in that protocol. A little bit of CoQ10 and some HIIT training, 20 minutes, 3 times a day, and we even told him how often to do. I think it was like 3 minutes. Low intensity, 2 minutes high intensity, 1 minute sort of low intensity again. And we gave him a very staggered protocol for these 20 minutes to do based upon the literature. Sometimes, Gail, you've ordered a lot of these tests. You may have seen intermittent fasting recommended. You may have seen intermittent fasting recommended with specific schedules, right? A 16-8, 21-4. We do give these based upon our research as well as the research that we've collated into our database. So we're able to give very specific things. We did that for his exercise. We also put him again on the supplementation. And we monitored him regularly. So what we saw at pre-treatment was that he had 80% enrichment of short-chain fatty acids. So they're just building up. His levels of these short-chain fatty acids were incredibly high. At post-treatment, that went down to 60%. I think this is cut off, but this is 3 months follow-up. We got him down to 40%, so half, roughly, his total enrichment. So the exercise, the supplementation, the dietary changes, these are helping. When we look at his lipid metabolism, and again, we have metabolites that underlie this, but health doesn't happen at a one metabolite level. Health happens, or disease even, happens at a pathway level. So now we're looking at his lipid metabolism. His percent enrichment was 45. At his post-treatment, we had 50. We took him off of this medication between the post-treatment and his 3-month follow-up, and he's at 75. So he's still going. Our goal with these protocols, and Gail, you and I have talked about this, is not to have people on supplements forever. It's to create a sustainable metabolic circuitry that can endure when these supplements have been removed. Nobody should be on 12 supplements. The guy who I mentioned earlier was on the cover. Huh? Unless you're selling them, which we never will for obvious reasons. So the guy, for instance, who was on the men's, I think it was Iron Man magazine, his issue was that he was taking, and I shit you not, 75 supplements, not every day, but weekly 75, right? And so I explained to him, and he was a physician, so I prepped some special slides for him to really explain to him what we saw, because I knew this guy is hell. He looks like the standard definition of health, and here I come, this chubby smoker on a call with him, and I'm telling him why he's not healthy. So I wanted to have the data ready, and I brought up some pathway results that aren't normally available. And he goes, and I say, what we're seeing is metabolic saturation. We're seeing things that are just built up. What's going on? And then he goes, I kind of thought that might be the case too. I'm on 75 supplements. Not every day, but throughout the course of a week, I'll take about 75 of them. And I'm like, this is not okay. Like, just eat a steak, have some, and his beta oxidation, so this individual, going back to John, 50% beta oxidation. So his throughput through this pathway was 50% what it should be for an age and sex match control with no evidence of disease. After our treatment, it went to 55%. Once he discontinued and retested in, I think, 3 or 4 months, I forget because it's cut off, it's 70%. So we see that his short-chain fatty acid levels dropped 20%, his lipid metabolism went up 5%, and his beta oxidation went up 5%. After discontinuing the medication, the protocols, that trend continued and in fact increased. We had similar decreases in his short-chain fatty acids with even more gains in lipid metabolism and beta oxidation. Okay, so this is all I'll say on workflow management because I am not a money doctor. I'll let the money doctors talk about how this works, but how we see our tests fit in is that a patient comes to you, you are the physician, you take the sample, you send it back to us, and that's it. We handle the rest. We will acquire the data, we will run that through predictive modeling and processing methods, and you still get this. You still get a scientist looking at your protocol. You still get Greg and Denver and Carmen, who are PhDs and trained in molecular omics, to consult with you. But when we do these predictive analytics, and this figure was made a couple years ago, so I'm sure AI is now faster than 120 to 240 times faster. But we are able to predict the health outcomes, so we are able to see this person's trajectory using some underlying equations, which we will get into at the end of this talk. We're able to do domain analysis, as Alex was saying. How do we turn this kind of overwhelming, unapproachable set of data into an easy to understand 1 to 10, right? And then how do we then generate a protocol based on that? So it's a model whereby the AI is in the center and it's interacting with a bunch of people, and the 3 people on the outside are our scientist, you, the physician, and the patient themselves. And it's a collaborative sort of effort between us to predict their health, tell them where they're at, and how to get where they want to go. Again, I won't belabor this point either. So this is the test collection, right? You've all taken it, I think, at this point. You send in the sample. That sample goes to our GCMS. We monitor 126 metabolites. Pattern recognition and AI black magic happens. And then what we're looking at is this health gap. We are looking at where this person could be, right? That's the first step in digital twinning, is looking at, I can tell someone this is how you lose weight. I don't know if I have many protocols for helping them gain like 6 inches of height, right? So if this was weight loss, maybe for me it could be a little bit higher, right? If it was, how much height can you gain in 3 months, it's going to be the same, right? So the first step in digital twinning is actually identifying where this person could be. The difference between where they could be and where they are, yeah.
How do they do this? I mean, like, they will take somebody and they don't know this person. I mean, like the machines which you have. And basically what will happen, that then it will tell you, oh, you can be here, but how it determines that's where you can be?
Great question. Yeah, so we utilize a very large set of data that comes from the Human Metabolome database. It's a database that was created after the completion of the Human Metabolome project. And that is, there's some biases in that data set with regard to geography and ethnicity, but we are able to match you to age and sex matched individuals with no evidence of disease. Or for instance, if we see persistent issues in phosphorylated metabolites, we might think that you have a certain genetic variant that enhances kinase activity. So that is something to consider. So we do consider those issues when we talk about, do we give this person a supplement, or do we recommend that they have the micronutrient through food? Because if they cannot digest the supplement, supplements are convenient, right? We know the dosages, but some people cannot digest them. So it is actually more beneficial to have it through the food. So when we can do those, we have this metabolic pattern, we're able to infer some sort of pharmacokinetic or pharmacodynamic relationship. We'll say that this person, and if they're reporting that they have cancer, obviously that this where they could be is going to go a little bit down, right? If they have certain predispositions, if this trajectory, right, this is on an individualized score level, but we could look at, pretend this line is methylation, right? If we know that they're homozygous for detrimental MTHFR mutation, this methylation potential is going to be a lot lower, right? And so the goal then becomes, how do you engineer them to get up there? First step is finding out where they could be. And we're improving on how we do this, utilizing demographic data, performing enrichment analyses, looking at, obviously we're able to profile 4,598 different SNPs using just these 126 metabolites. We're able to look at dysfunction in 970 different liver enzymes, mostly liver and mitochondrial enzymes, and say, okay, this person cannot handle certain kinds of drugs. They're going to be a very poor metabolizer of it. So their liver score is probably not going to be super, super high, right? We'll temper that. The algorithm will naturally temper that. And then the difference between where they're at and where they could be, this is going to inform the health recommendations. Not only are we trying to simulate these effects, but how do we know when to stop? How does the model know when to stop generating? This is in statistics is known as the model is converging. So the model has to know, I'm going to generate as many beneficial protocols under this domain as it takes to get this person up to here. There's no point, because we have 1,300 different interventions that we can give you, right? I recently shared the document with an ASU professor who requested it, and they were blown away by what we had curated. So the model converges and stops making recommendations for that particular domain when it thinks, okay, we've reached where this person can be in this state. There's no point in inundating them with 36-step protocol, right? Some dietary changes, some supplements, that is usually enough to keep them busy and adhere to a protocol. And adherence is inversely linked to number of steps, right? So we know this from clinical research, where the more you give them, the less likely they are to do. So we have to kind of balance the two using our own ability. So we set the model to converge when that person's score has basically gone as high as possible. And when we get to we're going to open up the digital twinning in a little bit and really look at the equations and underlying things that are happening in another case study. But these are usually based on 3 to 5 different things that are underlying this person's health. And they're not related to domain scores. They're related to things like lipid peroxidation. They're related to things like diminished ETC throughput. These are the factors that, if you address, will alleviate and assuage all of these poor scores together. And we'll show that in another graph where you'll see these lines, they'll converge onto certain factors. And so many of these health predictions, they share similar equations, because your environmental toxicity, your mitochondrial health, and your liver score is low because you have heavy metal toxicity. It's going to affect these 3 domains, right? So there's underlying factors that affect them as well. Okay, so how it all comes together, right? So you know this. You send in the sample. It usually takes about 2 to 5 days to get to you. You take it, 2 to 5 days to come back to us. This is what it looks like to be a physician who works with us. We do about a 2-day data processing step, 2-day data collection. We go through very advanced analyte ID using, of course, chemical standards. And then we have multiple data quality controls in step, both for handling, processing, and data collection off the mass spec. We then perform our analytics, our pattern recognition. We do disease enrichment. We have this unbelievable computational load when doing this. And then we do our in silico permutations. We try to do this in 24 hours, if at all possible. Sometimes it requires a little bit more elbow grease. And then you get on our sort of telehealth calendar. You schedule a talk with Greg or Carmen or Denver, and they have prepped for this 30-minute one-on-one, right? I think a lot of companies, they do a real disservice to themselves by entrusting a bachelor's degree kid. He's 2 weeks out of graduation. They put him in a 2-day training course, and then they put him in front of customers to discuss their real data. And that's pretty dangerous. So we have only PhDs, MDs that consult with you, and it's 30 minutes, and they prep for that ahead of time, right? So they prep some materials, some enhanced guidance. They look over the reports. They look over some pathway effects that you may have questions on, and they come with a wealth of experience. So all of this is kind of detached, right? Like the sample collection is detached. The analysis is esoteric. The machine learning is just hard to fathom. But then it comes in this report that I think a lot of people can read and digest, right? And the more you go into the report, obviously, the more in the weeds you get. But ultimately, it is approachable. And then at the end, you get this human touch, a human face that explains and is willing to work with you to digest those results. So here's an individual that we use as a good case study. John Doe. He comes to us as a 65-year-old retired dentist, DDS. We actually saw that he had heavy metal toxicity. We didn't know what heavy metal. Sometimes we can't. Sometimes with iterative testing, we get better as the model learns longitudinal data, but we had no idea. However, because as Alex was alluding to, we have also a Bayesian framework in place, the prior probability that it would be mercury poisoning was astronomical when you consider the fact that he's a dentist. He didn't come to us saying, I'm scared of mercury poisoning, right? He came to us saying, I have mild cognitive impairment, I have tremors at rest, I have difficulty concentrating, I'm always fatigued, and I'm worried. And I haven't been able to get to an answer. When we looked at his results, we just saw heavy metal. It could be lead, it could be mercury, it could be cadmium, it could be arsenic, tons of things that are under that domain, and none of them was significantly more enriched than the other. So they were all equally likely. However, when that prior probability gets updated with the fact that he's a retired dentist, suddenly it becomes clear that this is due to handling dental amalgams, especially when he was practicing, where PPE was kind of a secondary consideration. So when he came to us, he had 20% liver health score, 30% toxin exposure, which is bad in our case. And his mitochondrial health was at 30%, just looking at his TCA cycle metabolites and adenosine. So this is his baseline. His digital twinning recommendation, so he came to us with a physician, and it was one of those instances where the digital twinning model was allowed to prescribe some medications. We have over 100 medications whose effects we have also modeled in various dosages. We don't give them out, but in this instance, we did. We recommended, I think, DPCA, dimercaptosacinic acid. We recommended it at like 1 milligram per kilogram body weight. We recommended that he do 3 days on, 3 days off for the first 2 weeks, and then weekly blood pH testing. We want to make sure that the chelation agent doesn't change his pH. As I'm sure you guys know, pH is incredibly important for determining enzyme activity. You can completely make enzymes turn on and off by just changing blood pH. So making sure that the chelation therapy wasn't posing a metabolic challenge to that. We increased his intake of sulfur-rich foods, recommended things like specific amounts of garlic and broccoli in his diet. This is, of course, going to bind heavy metals as well. And in addition to that, we recommended lots of specific dosages of berries and timing of berries, specifically right before dinner, right before he went to sleep, to really aid in the natural detoxification and antioxidant processes that happen at night. These are circadian rhythm controlled pathways, so it's important when he's taking it. He did not have, a lot of people actually have this, is glutathione synthetase deficiency. It's super common. It's really underreported because it's not tested for. But we did not detect glutathione synthetase deficiency. This goes back to your point. It's like, where could he be? So we're not going to give him NAC, right? In an individual who has this deficiency, we're not going to give him NAC. He's not going to create glutathione. So he did not have that mutation. So we gave him a glutathione milk thistle. We gave him, glutathione precursor. We also gave him milk thistle to sort of increase the rates of hepatogenesis and hepatocyte turnover in the liver. Very specific dosage. John was a man who enjoyed his whiskey. We told him to limit that as much as possible, especially while the liver gets back to full speed. And then increase aerobic activities. John was a very bulky individual, and he did lots and lots of strength training. But when you have heavy metal toxicity, the focus really needs to be on aerobic activities, right? Cardiovascular health, on sweating. So we recommended that. He did have an infrared sauna. We recommended 20 minutes 3 times a week, infrared sauna, and increased hydration of, I think, more than like 3.5 liters a day. So this is his protocol. It spans everything from hydration to lifestyle changes like exercise and sauna usage, Very specific changes in his diet, not much. We're not asking him to become ketogenic. We were literally asking him to be mindful about consuming this amount of broccoli and garlic to get the necessary sulfur, and to be mindful about having a bowl of berries before dinner, right? These are really sort of innocuous changes at first glance, right? But never underestimate the cumulative impact and synergistic effect of these small changes. At 3 months follow-up, of course, he worked with his physician to get the prescription for the chelation agent. It's the only thing that required prescription. He emailed us within 1 week reporting on really improved levels of his energy. Cognitive function, so he had this sort of mild cognitive impairment, was over. Significant improvements, as we can see, at 3 months. His liver score went to a 6 out of 10. His toxin exposure score went to a 6 out of 10, so now he's normative in these domains. His mitochondria bounced from a 30 to a 9, so a 3 out of 10 to a 9 out of 10. And so, of course, he was ecstatic. He had felt like he got his life back. His son was actually a nutritionist who's now one of the healthcare providers on our platform and orders this for his practice. Okay, so this is a really recent case study. This happened this month, actually. So this is a 62-year-old Hispanic female. She's actually a retired OBGYN here in Scottsdale. And these are her actual survey responses, okay? I thought these were brilliant. Idiopathic musculoskeletal disease, GERD, chronic constipation, bipolar ADHD, PTSD, hypertension, diabetes, type 2, tergium keratoconjunctivitis. I think this is misspelled. It's just conjunctitis, right? Conjunctivitis, okay. And insomnia and microcytosis. Okay, cool. She also said, I take brand name, not generic, all of these. I mean, look at the litany of things she's on. Metformin, Percocet, huh? Yeah, Mounjaro. Restasis drops, melatonin, retin A, 0.1% cream, NAC, alpha lipoic acid, spermidine. She's going, I mean, this is a litany of things, and who knows how many of them she needs. I mean, she's taking things like spirulina and chlorella, and her environmental toxicity is a 1. So she is carrying this incredible toxic burden despite all the things she's doing. So she's throwing everything at the wall and it's not working. What you see in red is her baseline score. What you see in green is her follow-up. So we see significant improvements in all of these domains except gut, which was a 7, remained a 7, and her inflammation, so her mitochondria, which she was doing really well on. It's a 9 and went down to an 8. Okay, so she took a little bit of a hit there. So this is a Gantt chart showing everything that we found in her. So her test date was February 4th, right? Her recommended test date for retesting was May 4th. She didn't test when recommended. She retested at June 6th. And then this is her new recommended test date, which would be December 6th, okay? So what do we see? For aging index, we saw aberrations in polyamine synthesis, decreased spermidine levels, elevated oxidative stress. At retest, we still saw this persistent dysregulation of polyamines. We saw increased levels of N1 acetylspermidine and aberrations in NAD plus metabolism. So one thing persisted, and the top problems for her second and third issues related to aging changed. And so this is us interrogating the digital twinning platform, saying, okay, give me a Gantt chart of this individual with their #1 LLM handle, their large language model handle, which we use to classify these things. Give it to me, and then tell me what it was ranked as. So nutritional index, dysregulation in ketogenesis, low beta hydroxybutyrate levels, decreased magnesium and potassium. Okay, so we got rid of 2 of these. One of them persisted in low beta hydroxybutyrate levels, aberrations now in omega-3 fatty acid metabolism, and elevated markers of inflammatory cytokines. Inflammatory pathway. We saw aberrations in NF-kappa beta, we saw elevated TNF and IL-6, and decreased glutathione. So all of this changed when we looked at, so we addressed the issues in the inflammatory index, but we have new issues now. They're less serious issues, but these are the top 3 new issues that were found at retest. Persistent elevation of inflammatory cytokines, aberrations in COX and LOX, and elevated PGE2. Mitochondrial health, same thing. Dysregulation in the electron transport chain, decreased CoQ10 levels, and elevated ROS. At retest, we saw dysregulation in TCA, elevated mitochondrial DNA damage, and this is a really significant issue. So this is, her mitochondrial health is kind of going down, right? It went down at retest. And then we also had persistent mitochondrial dysfunction, which was actually getting worse. Cardiovascular health, aberrations in lipid metabolism, elevated LDL and triglycerides, decreased HDL. So we fixed some of these, except now at retest, we had this lipidemia was persisting. However, the model picked up now aberrations in magnesium and taurine. So you can imagine what those recommendations might be. And then elevated homocysteine. So we don't have homocysteine in our panel, but we get a very, very sensitive read on it by using methionine and cystathionine. Reproductive health. Nothing really to worry about. Focus on overall well-being. When you see this in a protocol, that literally means it's too much to give. This isn't our main worry, and there is probably some other recommendations given in those domains that are addressing this already. Don't worry about it. But at retest, we saw aberrations in folate metabolism. Elevated homocysteine is now an issue in both cardiovascular health as well as her reproductive health. So it's the third most important issue affecting her cardiovascular score at retest, and the second most important for reproductive health at retest. And then this dysregulation of omega-3 fatty acids, which was, again, the number 2 most important issue underlying her nutritional index score, which was improved, but still could have some ways to improve. So this goes on and on and on, right? We have aberrations in phase 1 and phase 2 detoxification. We can differentiate between the two. So if it's a CYP issue or if it's a conjugation issue, we can look at that. Elevated ALT and AST was inferred, and then decreased glutathione. Persistent liver enzyme elevation, cytochrome P450. So phase 2 metabolism is no longer an issue now. So at retest, her liver health improved. We had phase 1 and phase 2 issues, and now we only have phase 1. So the conjugation pathways were now working fine. Elevated bilirubin was also detected. Here, gut health. We're looking at dysbiosis. We can infer some elevated levels of zonulin and then some decreased short-chain fatty acid. Unlike the other person, we did not profile short-chain fatty acids, but we inferred them based upon levels of other fatty acids present. The dysbiosis kept going. We got rid of the elevated levels of zonulin, and now she only has elevated inflammatory markers associated with the gut and some decreased butyrate levels. So we ameliorated the profile of 13 short-chain fatty acids, and we only had butyrate now to contend with at retest. Neurocognitive. Omega-3 fatty acid pathway was an issue no longer in neurocognition. It's now an issue in reproductive health and down here in the nutritional index. So it's an issue, but it's not the first most important issue in neurocognitive health. It's now second and third in other domains. So what should be kind of apparent is that these domains are contingent upon underlying biological factors. That we can address these biological factors and move the scores of many domains up at once. Going on, aberrations in collagen synthesis, decreased vitamin C, elevated oxidative stress. Persistent dysregulation in collagen, so something's going on. Elevated markers of glycation end products. And we also have decreased hydration now. So she now is having low levels of metabolites that are associated with hydration. Okay? This is obviously going to affect integumentary health. Environmental toxicity, elevated levels of heavy metals, lead, mercury. This is saying that the model cannot detect. The model does not know what's going on. Persistent heavy metal burden continued. Aberrations in detoxification pathways, she had that also affecting her liver health. So these two are usually in lockstep in many people. Their environmental toxicity and liver health are going to suffer or benefit together. And then she had elevated oxidative stress markers. We know that her inflammatory index was absolutely horrible. That shouldn't be a news. The heavy metal persisted. We had aberrations glutathione and then elevated lipid peroxidation. We then had elevated oxidative DNA damage markers. We had deregulation in DNA repair pathways. So a lot of times these go hand in hand because those DNA repair mechanisms are contingent on low oxidative stress. As oxidative stress goes up, correction of nuclear mutations goes significantly down. And then she had increased OHDG levels. We had the persistent DNA damage at retest. Now we have aberrations. So the deregulation of the repair pathways have been largely fixed. And the second most important problem are these aberrations in P53 signaling. That's what's not giving her a perfect score in this domain. And then elevated levels of these DNA adducts. That goes back to the glycation issue we had. So what were the recommendations? Yeah?
I was just going to ask this. Whenever you see high homeocysteine, do you ever check the salt?
The what? Systol?
The sol, S-adenohomocysteine?
Oh, yeah. Acetylhomocysteine? No. We look at SAM, acetylmethionine, but not.
But part of the methylation cycle, if the homeocysteine is consistently high, a lot of times it's trying to go counterclockwise and hand that off to sol.
No, we haven't. And like I said, we don't look directly at homocysteine. We use methionine and cystathionine, which give like 98% accuracy to homocysteine levels. No, I haven't looked at that. So homocysteine, if it's elevated, you're saying it goes to SAH?
Yes, because it can go counterclockwise in the methylation cycle.
That's really interesting. No, I haven't looked at that. No. Usually when we see elevated homocysteine, we're thinking to ourselves, okay, is there an issue with methionine synthase? Is there an issue with methyl groups in general? So looking at their sarcosine levels, which is N-methylglycine. Sometimes we'll look at metabolites that tell us if there's a methionine synthase deficiency. And we can, again, the ratio between methionine and homocysteine is actually more clinically relevant than just homocysteine levels. So when we look at like the predicting cardiovascular risk in 5 years and 10 years, that ratio of methionine and homocysteine is more valid. But I'll look into the SAW.
Yeah, I've read where a lot of people think sol is actually more accurate than homeocysteine.
Oh, well, that's right up our alley. We'll definitely have to take a look at that, especially when they start talking about accuracy and prediction. That's incredibly relevant to us. I'll look into that for sure. So how do the recommendations for this individual change, right? So spermidine supplementation at her first baseline, we recommended 1 to 2 milligrams a day, omega-3 fatty acids, 1,000 milligrams twice a day, and mindfulness, yoga and Tai Chi, seriously. Now at retest, we're like, okay, your spermidine's going up. Now let's cut it down to just 1 milligram a day. You don't have to go 1 to 2. Your niacinamide's a little bit low, so let's address that with 500 milligrams twice a day. Palmitic acid, this is usually from fatty cuts of meat that we want to limit. That was her primary concern. Nutritional index, we recommended keto-compatible, high-nutrient foods. This is where we try to be sort of respective of an individual's chosen diet, right? So you've taken the surveys, you've seen that there's a portion of diet. So if an individual is vegan, we're not going to recommend fish oil, we'll recommend algae oil. In this case, she was adamant that she wanted to stay keto. So we gave her keto-compatible but high-nutrient foods. A lot of times these individuals that are in constant ketogenesis, they're not eating all of their macros and micros in the ratios that they should be. So we designed a protocol for her with that, supplementing with bone broth 2 cups a day, and then multivitamin, magnesium, and supplements at the RDA. No customization there. Increase whole grain consumption to 2 servings a day. So at retest, we can get rid of all of this. We wanted to increase her whole grain consumption now to 2 servings a day, and we were like, listen, you have to have some of this, okay? You can't just be all ketogenic, and hopefully she adheres to that until December 6th. And we also said, okay, the 1,000 milligrams of omega-3s, let's go to 2 grams now. We're gonna modulate that because the issue that we saw in the nutritional index was related to this. Curcumin supplementation, 500 milligrams twice a day. We kept that the same. And then glutathione boosting NAC. She did not have synthetase deficiency. We kept that the same. So some of these recommendations aren't going to change, right? If they've worked and they're predicted to improve the trajectory of the underlying factors until retest, they'll stay the same. Same thing here. CoQ10, 200 milligrams a day. Now we're going to 200 milligrams twice a day, right? A little boost in the CoQ10. We also want to do that because we saw her mitochondrial score take a hit at retest. Okay? Alpha lipoic acid, 600 milligrams a day. We went from reducing saturated fat intake, fiber-rich keto foods, and red yeast rice, 900 milligrams, to just switching it totally. She had magnesium and taurine issues, so we're now going to give her 400 milligrams a day of magnesium and 1 gram of taurine a day. Same thing here, no direct interventions, was focused on overall well-being. Now we're going to say, okay, we're going to take methylfolate at 800 micrograms a day for your elevated homocysteine, which we saw. So we want to give methylfolate, so we avoid the folate trap, right? Methionine can hand off that methyl group to homocysteine, tetrahydrofolate can hand off that methyl group to homocysteine and regenerate methionine. Where are we at? Right, and then omega-3 fatty acids, again, 2 grams a day. So they're always the same. Cruciferous vegetables, milk thistle, 150 milligrams twice a day. Let's boost the milk thistle. Now it's the #1 recommendation at retest, 200 milligrams twice a day, and reduce the alcohol consumption. Because again, we're seeing she's retired, she likes wine, and we're seeing those aldehyde metabolites are now the #2 issue. Maintain current practices and diet. Okay, no changes for this domain at the first baseline test. At retest, we're giving her a very low dose of probiotics, 10 billion CFUs, and we're increasing her fiber intake to 25 to 30, right? Like, what's the recommendation for fiber? I just saw this. It's like 40 grams or something. Yeah, yeah, right? But when we look at the science, we see that people actually need like 50, 60 grams of fiber, right? So even the recommendations are well below what they should be. And at Therione, we don't have to go by the recommendations. We can use the science. So omega-3 fatty acids, we're at 2 grams a day. Phosphatidylserine, 300 milligrams a day. Curcumin, 500 milligrams twice a day. Mindfulness meditation and cognitive behavioral therapy. These are recommendations. This is because this person came to us with ADHD, bipolar, PTSD, right? Did you see the litany of things that she had? So this is where the metadata comes into play and say, listen, I see some neurocognitive issues, but I'm also getting metadata that's telling me cognitive behavioral therapy and mindfulness and yoga and Tai Chi, these are all really important things for an individual who's suffering from the myriad of issues that she reported. At retest, we just said, okay, continue the omega-3 and CoQ10, as stated down here. We're boosting the CoQ10 a little bit. And then instead of doing this phosphatidylserine 300 milligrams a day, let's break it up, 100 milligrams 3 times a day with specific times. So now we're trying to, we've given her body this incredible boost, and now we want to sustain that with a regular pool of phosphatidylserine. Hydration, antioxidant rich for her skin, hair, and nail health. Hydration, antioxidant rich diet, topical vitamin C, topical, gentle skincare routine, and sun protection. So we have metabolites that tell us things such as, epithelial cell death due to UV radiation. That's a really fancy way of saying sunburn, but we have a model for that. We have a signature for that. So we can recommend things like take an SPF 30. It's really important. Like this is seemingly a very superficial recommendation, but it's going to have significant gains. As we saw, however, if you remember from the slide before, her main issue at retest was now persistent dysregulation in collagen metabolism, which was also detected at her baseline. And now she has elevated markers of glycation end products and decreased hydration. So we're going to ameliorate 2 of them here with the collagen peptides, 10 grams a day. And you can't make collagen without vitamin C. So 1,000 milligrams a day, that's the ratio that is reported in the science for generating the amount of collagen that we wanted to get to her. So that's an instance where the digital twin model is saying, okay, a collagen production for this person should be about here. We're going to give them this much collagen, but we're also going to boost endogenous collagen synthesis by supplementing with vitamin C. These 2 should have the effect sizes we're looking for. Yeah, this is all AI. This is all AI. Even here, even here, this is all AI. So this is AI saying, okay, I'm interrogating this patient's records, right? I gave it the data again, and I said, okay, I want to do this. So we can essentially feed this thing code for this Gantt chart and say, here are the timelines, here's the data structure. Using statistics, match it to the large language model handle, and then kick out what was identified as the #1 issue for her. We don't do this for everyone, right? Like we do this when we see a really interesting case, and we're like, what happened? How did digital twinning adapt to re-identify the most pressing issues at retest, predict her out 6 months, and then give these recommendations? So the environmental toxicity was really great. So we had environmental toxicity. She has elevated heavy metals, lead, mercury, we don't know. Aberrations in detoxification, elevated oxidative stress. What do we do here? Okay, we were like air purifiers, detoxifying foods like cilantro. We also want you to take a look at your household cleaners. They might be something in there. Do an audit of that. And then regular exercise, because that is the #1 way to easily move the needle on these things. Broadly speaking, whether it's lead or mercury, it doesn't matter. Exercise will help. But then at retest, when we saw that it was a persistent heavy metal, definitely heavy metal now, we're sure of it. We see also aberrations in glutathione and elevated lipid peroxidation. Now we're saying, okay, time for you to take that NAC, 600 milligrams a day. Also, we want you to take activated charcoal, 500 milligrams twice a week.
When you're trying to get your precursors for glutathione, do you ever give glycine and glutamine along with NAC? Yeah.
Absolutely. So a lot of times we'll give glycine if glycine is low and they don't have a synthetase deficiency. Glycine will be a recommendation. Glutamine we give, most of the time we give glutamine as a repair for gut lining. So we really like the glutamine. The data are pretty yeah, there's a preponderance of evidence that shows that glutamine is this really like, for most people, it's like a silver bullet for leaky gut issues, or I don't know, leaky gut isn't really a thing. It's like a cluster of symptoms. But yeah, so absolutely we do. Yeah, exactly. And then for her mutational load, right, we had some antioxidants and polyphenol-rich foods. We saw low levels of metabolites that are related to sleep quality. So we were like, okay, you got to focus on sleep quality. Keep in mind that at night is when these DNA repair mechanisms work. When right now your repair enzymes are not focused, or they're not working, they're not active. They're timed to circadian rhythm. So as you sleep, cortisol levels go down, and these things shoot up and they start activating. So we were like, okay, you have to have some stress management and some better sleep. Also, I mean, given her family history, I didn't put that on the thing. She had lots and lots of family members with cancer. So one way to stay healthy, especially if you have a familial history of cancer, is to get regular checkups. Regular checkups are positively correlated with improved prognosis, even without a diagnosis of this person has cancer, right? That is a great screening method. Then at retest, we were like, okay, you actually improved a little bit. So what are we going to do now? Vitamin D3, we're going to give you 2,000 IU a day, relatively standard. We also want you to take this green tea extract, 500 milligrams a day. So this is how the model, again, matches, first identifies issues, corrects them, and at retest says, okay, we're not just out here to identify new issues. We want to know what issues were resolved. Knowing what issues were resolved is going to tell you a lot about how this person's metabolism is working. What treatments that we gave were they responsive to? If they were really responsive, shit, give them twice as much. If they weren't responsive, or if now we're certain of something that we were kind of hesitant about before, put them on a chelation protocol. So this is how the model begins to learn. Now, this is the actual equations that underlie this. And what you can see is, for all of these conditions, right, so she started off, her overall health score was a 30 at baseline, and it went to a 62 at retest. So these are 5 factors. We're going to talk about factor analysis in the next talk. But these are 5 factors that underlie her health. Each factor, you can tell, for this factor only has 1 node going through it. This factor has 3 nodes going through it. This factor, 2 nodes. This factor, 3. This factor, 2. And what it's telling us is that there are 5 total metabolic processes. In this case, I think one was NF-kappa beta, one was lipid peroxidation, one was persistent mitochondrial dysregulation. I forget the other 2. But these are factors. They're not necessarily biomarkers, but they're trends. And these trends, depending on which trends they are essentially the nodes that will determine this person's trajectory at retest. So by identifying these factors and treating them with our protocols, we now know that her equation is great, right? We also know that we're gonna run this gambit here with the mitochondrial health. She's gonna probably go down another point here. Even with that extra CoQ10, her mitochondrial health is gonna go down to a 7. We predict, right? But the model is also good at doing sort of a cost analysis, right? What we have is what's called a MDA, mean decrease in accuracy. And sometimes we have to give up some issue here so that these other trends can have improved outcomes, right? So it's running a chess gambit here. It's sacrificing a pawn so it can move some other things up. And I'm not saying that mitochondria are not important, but she's going from optimized to still optimized to on the high end of normal still. We're not risking her to be in a poor metabotype. She's not in the lowest metabotype. So you can see that, for instance, some of these factors, like this one here, is controlling the outcome of 3 trajectories. 3 scores have to be addressed by this factor to improve. These are the nodes that underlie the health scores. So we can do this for every individual. We don't because it's incredibly time consuming and we don't have a pipeline for it yet. But this is essentially where we see her at, right? So we know that her gut health is going to stay the same, her inflammation is going to go down, but we are certain that, at December 6th, assuming roughly 85% adherence to the protocol, we're going to go from an average of like somewhere around here to an average somewhere around here across all scores and domains. And this is the underlying math of the digital twinning. This is saying, here are the underlying factors, the pathways that are controlling more than one pathway, that are determining the outcome of more than one score. Scores are scores. We come up with scores. Okay, we validate them with the literature. We do some statistics. It's very fancy. But the issue is we want to address the underlying concern, and this is essentially what's going on. And I should have probably labeled it and said this one is lipid peroxidation and this one is mitochondrial dysfunction, but you understand. And we'll get into factor analysis more with the next talk.